Hey, good evening, everyone. Hope you guys have been doing well and you've had a good fall break last week and are ready for chapter 13. This chapter talks about meiosis, which is basically talking about how uh, do the body produces gametes, which are basically cells that are required for reproduction. Okay, during sexual reproduction. Okay, so we're gonna start with that. As you may know, the sexual reproduction involves a sperm fertilizing an egg. Okay, so it's always one sperm and one egg that are being used to to reproduce. Okay. So, this is a busy slide, but this is just showing you meiosis, which is the process that cells in the case of like the male, the cells produces the um, sperms, and these would be basically produced in the testes of a male, and then in the female, the eggs are produced from the ovaries. Okay, so this is what you're seeing here. So the father's testes is on the left side, and the mother's ovaries on the right side. Okay, so the way all of this works is that humans have 46 chromosomes in total. Okay, so 23 chromosomes are normally from the father, and 23 chromosomes are from the mother. Okay, um, so so the meiosis involves several steps. So that there's two parts basically. So you have meiosis one and meiosis two that give you finally the products of what we call um, haploid cells. Okay, so basically the final cell, the result of meiosis, produces instead of 46 chromosome, you'll have 23 chromosomes in each cell. Okay, so if we're talking about a sperm, you'll have 23 chromosomes in each sperm. And then if we're talking about the egg, you also have 23 chromosomes. So you can imagine if these two were combined, you'll have 23 plus 23, which is 46 chromosomes. Okay, um, and also just so I can tell you this right off the bat. So the 23 chromosomes in the sperm, okay, 22 of them are going to be what we call somatic chromosomes. Okay, so these are basically coding for everything in the body except the actual sexual orientation of um, you know of whatever embryo is gonna be reproduced. So you'll have basically 22 plus either an X chromosome, which is the sex chromosome, or a Y chromosome in the case of males. In the case of females, you'll have 22 somatic chromosomes and basically an X, okay, as the only possible basically sex chromosome, okay, because males have XY as their sex chromosomes, while females have XX as their sex chromosomes, okay, so they're only able to give that X. Okay, so um, when sperm and egg unite, we call this fertilization. Okay, so the sperm fertilizes the egg by basically entering the egg. Okay. Let me just tell you also right off the bat, in meiosis one, um, we have basically homologous pairs of chromosomes are basically produced basically, and then after they're produced, they're separated. So this is what you end up with. So this is right here, meiosis one, and then during meiosis two is when basically all the different sister chromatids are actually separated out. So right after that, you can kind of remember that meiosis 2 is kind of similar to mitosis, because mitosis involves the separation of sister chromatids. So this is a similar process here. Okay, so um, the basically the offsprings basically are resembling their parents, right? Um, because the genes are really like passed on from the parents. But there's obviously differences, and we will see how that happens, and what causes all the differences, from the like from a baby. That why does it look different than, like, you don't look the same as your parents, right? You don't exactly look the same. You have resemblances, but not the same. Also wanted to define heredity, right? So that's the transmission. Okay, it comes from hereditary. Okay, so it's the transmission of traits. Traits are basically like eye color, hair color, height, anything like that. Okay, so it's things that you can see, phenotype. Okay, from one generation to the next. When we say variation, basically, is the differences, right, in appearance that offspring show from parents and siblings. Okay, so when we say, like, for example, a baby has, like, let's say, um, you know, uh, black hair, while his parents have, for example, brown and blonde hair. Okay, so that's variation. Okay, so it looks different. Genetics is basically the scientific study of heredity and variation, both of them. Okay, and it comes from the word genes. So that's basically, genes contain all the coding information, and they're actually, what makes up basically DNAs. Okay. Okay, so again, they're the units of heredity, right? Heredity. So they have all the basically coding that codes for. So a gene might code for a certain eye color, and might code for certain skin colors, certain height. Maybe you know differences in voice. So all of these things are basically coded by genes. Okay, and they are made up of segments of DNA, as I mentioned. Okay. So do you remember how the DNA looks like in the nucleus, right? It's all like spaghetti-like type of deal, okay? And that's how it is before it's actually condensed into the chromosomes, right? During mitosis. So the genes are basically made up of those little segments of DNA, okay? And obviously you could imagine, since we know that DNA is made out of like different bases. Do you remember that from, like, we could say that it's got like a G base, a C base, um, you know, uh, uh, and the other bases, right? Pyrimidines and purines, right? These are the bases, and so these genes are made out of like all those different bases attached with each other. Okay, you find, like, just depending on the gene, you might find, like, 30 or maybe 100 or maybe 1,000 base pairs connect with each other. Okay. So genes are passed to the next generation via reproductive cells. So we're talking about what? We're talking about sperm cells. This would be a reproductive cell. We're talking about egg cells. Okay. These are also called gametes. Okay. So if I ask you, what are gametes, right? They're sperm and eggs. Okay, so sperm in the case of male, eggs in the case of females. So most DNA is packaged into chromosome, right? We know that because, during mitosis, the DNA is basically condensed into its chromosomal shapes. Okay? Outside of that, it's basically uncondensed or non-condensed, and, researchers have to basically, in order for them to, like, study chromosomes and look at the specific shape of chromosomes, they have to actually wait for mitosis to happen. Okay, so humans have 46 chromosomes in their nucleus. Okay, basically in their somatic cells only. Just know this one. It's important. So there are 46 total in each of your cells, except, okay, when we're talking about, reproductive cells, things become a little bit different. Okay, so somatic cells have 46 chromosomes, while gametes have 23 chromosomes, right? So why do gametes have, like sperm and eggs, why do they have 23 chromosomes? Because, they, during reproduction is when, a sperm meets an egg, you have 23 plus 23, as I mentioned, and you get 46 total, okay? All right, so, a gene-specific position right here along a chromosome. So, by now how a chromosome looks like, right? Let me see if I have, okay, anyways, um, I'll get to it in a bit. So, a location on a like specific pinpoint on a chromosome, we call this locus. Okay, just think about it: location, locus. Okay, so that tells you that we're asking about a certain location on that chromosome. Okay, so just looking at the differences between asexual and sexual reproduction. In asexual reproduction, right, as it sounds, there's only single individual, right? That's why it's asexual. It passes all of its genes to its offspring, right, without the fusion of gametes. So why? Because it's asexual, right? There's no, egg and sperm, so there's no fusion of different gametes, okay? Now, when we say clone, a clone is a group of basically genetically identical individual from the same parent, okay? So it's exactly, as we know it. When we say clone, you're cloning something, so you're making a copy paste of it, okay? So in sexual reproduction, right, two parents, right, male and female, right, give rise to offsprings that have unique combination of genes, okay? And we will see why later on in this chapter. How do, these genes become, different than the parents, right? Um, what causes all of these things? And this is just showing you, the asexual reproduction in two multicellular organisms. Okay, so the hydra and the redwoods, as an example. All right, so looking at the life cycle is a generation to generation sequence of stages, right? In the reproductive history of an organism. Okay. Um, So there are stages, right? In which the organism basically will become an adult and basically reproduce, and then the offsprings do the same thing. So that's what we call basically a life cycle. Okay. Uh, let's see here. Okay, so what you're looking at right here on the right side is what we call karyotyping. Okay, so karyotyping involves, actually displaying all 46 chromosomes of a human, right, on display like so, next to each other. You notice that basically we have basically 23 homologous chromosomes. Okay, so 23 pairs of chromosomes. Okay, one chromosome is coming from what? The first parent, and the second parent is given the second chromosome. Okay, this is how we have, remember, 23 plus 23 is 46. Now 22 pairs are what? Somatic, right? As I mentioned before, somatic. Okay, and then basically you have the last two, so it would be, so 44 total are somatic, and then the last two are the sex chromosomes. Okay, so these sex chromosomes are what's gonna determine if somebody is a male or a female. Okay, so if a person has an XY, it's a male, and if a person has an XX, it's a female. Okay, let's see here. So again, remember that two of these chromosomes, we call them homologous chromosomes, okay? So they're basically kind of the same length, as you can see. They look similar, don't they? But obviously, you know, they basically have different genes, but they carry the same inherited characters. Okay. So let's see here. So sex chromosomes, we said, you know, the last two chromosomes. So you have, you know, either XX or XY. And I went over all of this, and you know, you have your 22 pairs of chromosomes are autosomes or somatic chromosomes. Okay. Remember that. So 22 pairs of autosomes give you 44 total. Okay. And then basically you have two sex chromosomes. Okay, so as I mentioned before, so you have basically each pair of homologous chromosomes. You have one chromosome in each pair from each parent. Okay. Um, and we went over all of this. Okay. So all of these chromosomes are found in every single cell except your gametes. So except sperms and eggs. Okay. Just so you know how things you know, happen. Okay. So you have 46 chromosomes in each of your skin cell, muscle cell, nerve cell, whatever you want, except the gametes. Okay. So these somatic cells, okay, we call they're diploid cells, okay, which means they have you know, two sets of chromosomes. That's why it's defined as 2n. Okay, so if 2n is a diploid cell, then what would be n in this situation? Is n is equal to what? 23, right? So 23 times 2, you get basically 46. Okay. Okay. So basically, we know that, you know, anytime we want to have basically division happen, we need to replicate the DNA first, right? So we know that, you know, the DNA has to be synthesized. In the case of mitosis, you know, we knew that this is going to happen where in the S phase. This is also kind of similar, okay? So the DNA has to be synthesized and the chromosomes have to be replicated. Same exact thing, okay? So basically you have, you know, the parental chromosomes, and you have the mother chromosomes, okay, maternal chromosomes, and you have basically you know, sister chromatids. You know, after the chromosome is basically replicated, you get basically two sister chromatids, exactly the same thing, and you know, where it happens, you know, and how it happens in mitosis, okay? So each replicated chromosome, as you can see here, okay, consists of two identical chromatids, sister chromatids we call, okay? So now let's look at a gamete, right? A gamete, again, we said, what is that? We said it could be either a sperm or an egg, okay? Those contain a single set, as I told you before. So a single set of chromosomes. So instead of pairs, they're only one set, okay? And those basically have what, you know, an n equal 23 only. So we call them haploid cells, okay? So remember, gametes, sperm or eggs are haploid cells, okay? Now all the rest of our cells, like, you know, somatic autosome cells, like skin cells, muscle cells, you know, etc., they're all diploid cells. So all of them have 46, except gametes are haploid cells, and these have 23. Okay? So you have basically in those 23, as I mentioned, you got basically 22 autosomes, and you got the single sex chromosome. So, you know, it could be an X, it could be a Y. In the case of males, you know, that sex chromosome, it could be an X or an Y. In a female, egg, the sex chromosome is always just an X. Okay? And this is just showing you the same exact thing again. Okay? So we know that the fertilization is the union of gametes, right? Of both sperm and egg. Now, when an egg is fertilized, right, we call this a zygote by this time, okay? So the zygote has one set of chromosomes from each parent. Okay, how can we define this? So one set of chromosome from each parent. This means that a sperm is carrying 23 chromosomes and an egg is carrying 23 chromosomes. That would be one set from each, which equals to 46 chromosomes, okay? So now we formed a diploid cell here, okay? And so a zygote is made out of, is a diploid cell, okay? After we form the zygote, right, the cells starts proceeding with mitosis, which is regular cell division, okay, that we learned about in the last chapter. Okay, and that's how, one cell becomes two cells become four cells become eight, so you got the doubling keeps happening until, all the different, organ systems, all the different body parts, whatever happen, and, this zygote becomes an adult, okay? Okay, so let's see here. So we know that at sexual maturity, right, and the ovaries and testes produce haploid gametes. Okay? So the cells in basically ovaries and testes, are they haploid or are they diploid? The cells are diploid. Okay? What they produce finally is a haploid, and this is what we're studying today. We're studying meiosis, which is how do actually, testes and ovaries make a haploid, which is, let's say, a sperm or an egg from a diploid cell, okay? So let's continue here. Okay, so we said that meiosis results in one set of chromosomes in each gamete. Hopefully this is making sense by now, okay? So let's just take a look at this diagram. So we know that multicellular diploid adults, right, are regular cells, diploid cells are 2n, which is equal to 46, okay? Using the testes or ovaries, right, we have the process of meiosis happen, right? Meiosis produces the haploid gametes, whether it's an egg or a sperm, which is equal to n is equal to 23 chromosomes, okay? When fertilization happen and both sperm and egg is combined, right, we produce this zygote, which is a diploid cell, right? That's what I told you the slide previously. So this would be 2n is equal to 46. Do you see how the process is repeating itself? Now this goes through mitosis and development, as I told you, okay? To, basically multiply the cells. Okay, so the alteration of meiosis and fertilization is common, right? And all organisms that reproduce sexually, okay? The three main types of sexual life cycles differ in the timing of meiosis and fertilization. Okay, so we're going to learn about, how animals differ from plants, from like, fungi and some protists. Okay, so let's go. I know this looks very busy, but we'll go through each one at a time. Okay, so let's look at the animal, sexual life cycle first. Okay. So gametes, again, as you know, right, are the only haploid cells in animals, and humans too, right? So the rest of the cells are what? Diploid cells, okay? Basically, they're produced by meiosis, as we know this by now, right? They're produced in the testes or ovaries, depending on if they're sperms or eggs, and basically they don't go you know, no further cell division before fertilization. So, you know, a sperm is gonna stay a sperm, an egg is gonna stay a an egg, sorry, until, you know, they meet and fertilization happen. Okay. So, you know, gametes fuse to form the diploid zygote, right? And that zygote is gonna basically, you know, go through mitosis. As I mentioned, this is just repetition. Okay. So you can see that's just exactly the same thing we just went over. So, okay, so we start with the diploid, right? It's producing through meiosis the sex gametes, okay, which is, you know, just N here, right? When you have, for example, this N could be either a sperm or an egg, you know, meets or gets fertilized with, you know, another sperm or an egg, right? You have fertilization happen, which produces a zygote, which is 2N, and that goes through mitosis again. Okay. Now, in the case of, sorry about that, it's my daughter in the background. So, anyways, in the case of plants and some algae, we have a little bit of differences here. Okay, so this is why we call this an alteration of generation. Okay. So basically, its life cycle includes actually both a diploid and a haploid multicellular stage, and we'll see how that is. Okay. So what happened is here we start with basically a diploid multicellular organism. Okay. This is what we refer to as sporophyte. Okay. So the sporophyte actually, which is a diploid, you can see that, right? 2N. It goes through meiosis. And then through meiosis, right, we're producing 1N, okay, which is a haploid here, okay? Then, instead of, you know, going directly and, like, you know, meet another haploid and gets, you know, through fertilization, we actually go through mitosis again. You see that? So meiosis, then mitosis here, and then due to mitosis, okay, you know, we have a haploid multicellular organism, okay? We call this a gametophyte. Hopefully I'm pronouncing this correctly. Which this gametophyte is basically going through mitosis again. And what happened is, you know, if we have another basically, you know, gamete from another plant come here and they basically you know, meet this one haploid gamete, fertilization happens, okay, and then you have a zygote. So from here to here is kind of the same, okay, as animals. So you know, fertilization happens and zygote is produced. Then the zygote goes through mitosis again, you know, to go through the cell divisions, regular cell divisions, and it goes through the same thing again. Okay. So just so we can kind of like you know, understand this a little bit. So sporophytes comes from the word spores. Have you heard of that before? I'm pretty sure you've heard of that before. So these basically make haploid spores through meiosis. So this end right here, these two ends, they're called spores. Do you see that? Okay. And then the spores are basically, they go through mitosis and we get this haploid multicellular, so it's a haploid here now at this point, multicellular organism, right? And then this produces the gametes, so this is right here is where fertilization can happen, okay? It can't happen at the spores portion, it could only happen at the gametes portion, okay? So this is just explaining the same type of deal. So each spore grows by mitosis, right? As you can see here, into a haploid organism called gametophyte. Okay? Then gametophyte makes haploid gametes. Do you see that? By mitosis. Okay? and then your fertilization, occurs, and then we have a diploid sporophyte, which is this. Okay. All right. So now let's look at fungi a little bit. Okay. So basically, in most fungi and some protists, the only diploid stage is a single-celled zygote. Okay. So this is the only diploid stage, unlike here where we have like the zygote and this sporophyte. They're both diploids. Okay. So you don't have a multicellular diploid stage. Okay, because this is single cellular. Okay. So basically, the zygote in this situation, which is a diploid, okay, goes through meiosis, and then basically, we produce here a haploid. Okay, haploid cells. Do you see that? Okay, and then that basically, each haploid cells grows by mitosis. Okay, into a haploid multicellular organism, which is right here. Okay, and that basically produces that adult, okay, produces your gametes right here, which basically allows for fertilization. So anytime you see fertilization, you do need gametes. Okay. All right. So yeah, depending on the type of life cycle, whether it's like an animal, or a plant, or a protist, you can have either a haploid or a diploid cells that can divide by mitosis. Okay. So can, for example, let's see here. Never mind. I'll tell you a little bit about it. It's too early for it. But yeah, in all three life cycles, basically the halving and doubling of chromosomes contributes basically to genetic variation in offspring. Okay. Not only that, but this is just a summary, but we will see how actually there's a combination of genes that happen, not only just the doubling and halving, that actually allows for variation. Okay. So just know that only diploid cells can undergo meiosis. Okay? Why is that, you might ask? Like, can we have a haploid cell, which is made out of 23 chromosome, go through meiosis again? So we're talking about making this 23 chromosome, we're making it into 12.5. That's impossible. Okay? So only diploid cells can actually undergo meiosis. Okay? So, and I said this before, right? Exactly like mitosis, we need the chromosomes, which is the DNA, to be replicated, right? Before we can proceed to meiosis 1. Okay? So, meiosis have both meiosis 1 and meiosis 2 parts. Okay? We will study right now how different both stages are. Okay? So we know that from one um, sexual cell, okay, located in the testes or in the ovaries, okay. From one, we know that 4 daughter cells, their haploid cells, are produced. So they can be, in the case of males, there are going to be 4 sperm cells. In the case of females, 4 egg cells. Okay. We know that each of these haploids, they have basically half of the as many chromosomes as the parental cell, right? The cell that's producing them. That's why it's a diploid. Okay. So this is a diploid cell. When the process after meiosis 2 is completed, we have haploid cells. Okay, so chromosomes are duplicated before meiosis starts, as you can see here. So we have basically a pair of homologous chromosomes in this situation and a diploid parental cell. Okay, so one is from the father, one is from the mother. Okay? So this cell technically has 46 chromosomes total. Okay? Now, what happened here is that each one of these chromosomes is basically duplicated. Okay? And so you have sister chromatids. Okay? Before we have, sister chromatids are basically duplicated chromosomes before we proceed into meiosis 1. Okay? And as you can see, they're basically associated along their length. Okay? They look kind of similar. And so there's basically what we call cohesion in between them, okay? That basically allows them to be kind of attached, okay? Alright, so let's go through meiosis now. So meiosis 1 has basically all those 4 stages. So we could say prophase 1, metaphase 1, anaphase 1, telophase 1. So it's unlike mitosis where, you don't put the number after prophase, right? Here in meiosis, whenever you see a number after it, that means we're talking about a process in meiosis. Okay, don't get this mixed up. I know they are the same stages, and in many cases there are similarities, but there are big differences also between meiosis and mitosis, and we will see what that is. So let's just take a look a little bit here. So this is a summary of meiosis 1 you're seeing here on the left side. Okay, so we're looking at prophase 1, which is the first stage in this slide. Okay, so in early prophase 1, each chromosome pairs with its homologue and crossing over occurs. So what you're seeing here, right here in prophase 1, this is basically the cell, how it looks like at the end of prophase 1. Okay, so when we're talking about crossing over occurs, this means, is exactly, don't get it confused, it's just not that difficult. Crossing over, so there's things crossing over to the next side, okay? So the homologous chromosomes you see here, okay, they get basically attached, and I will explain how that happens in a bit. They get attached here at certain locations, okay, and they basically exchange genetic material at those locations, okay? And not only that, but they form those, where they exchange the genetic material, they form X-shaped regions, and these are called chiasmata, okay? So these are where the crossing over of different genetic information happens, okay? So this is actually very important, okay? This is why, primarily, one of the reasons, but I'll explain why. There's like 3 big reasons why, offsprings look different and completely different and actually unique from their parents, okay? Okay, so again, this is just showing you the whole meiosis 1. So you have metaphase 1, anaphase 1, telophase 1, and cytokinesis. Similar processes are happening after prophase 1. Similar processes are happening like mitosis, but there are differences, obviously, okay? So let me just explain the crossing over process here that we just went over, briefly. So basically, crossing over involves, the sharing of genetic information. So let's start from here, from the beginning. So after interphase, do you remember what interphase is? It's basically the stage, before, meiosis or mitosis happens. Okay, it's also the stage where the S phase is in, where the chromosomes are, or the DNA and chromosomes, right, the form that forms chromosomes, are replicated. Okay, so after interphase, the sister chromatids are basically held together by proteins called cohesions. Okay, so we basically duplicated the chromosome, and now we have sister chromatids. Okay, so let's see here. if we're looking at the red ones are maternal sister chromatids, and then the blue ones are paternal sister chromatids. Okay, so before the S phase, we only had one of these lines, one red and one blue. After we went through interphase, we have duplicated each one, and now we have double blue lines, as you can see here, and double red lines, as you can see here. Okay, so, and they are basically held together, each one, by cohesions. These are like basically rubber bands, and they're basically holding the sister chromatids together. Okay. Okay, so now what happened is, and this is done in meiosis, kind of like automatically. Okay, so the non-sister chromatids, as you can see here, they start having basically they broke at certain precise positions, and those precise positions are actually facing each other, which is pretty cool. Okay, so after this happens, okay, what happened is that the two chromosomes actually um, get close with each other, and they form this zipper-like structure in what we call synaptomal complex. I think I pronounced this correctly. Synaptomenomal complex. So these basically holds the homologs, both chromosomes, together tightly, as you can see. Okay. So during synapsis, so this is another process, right? DNA breaks, the ones that occurred here, they basically get repaired. Okay, and the way it gets repaired actually is that this red right here is joined to this blue here. Instead of repairing basically the blue to blue and red to red, they repair it this way, which the red is joined to blue and blue to red. Okay, so basically this is what's shown you here. So we have a joining DNA from one non-sister chromatid, so from the other side, okay, to the corresponding segment of another, okay, which is like, next to it, facing it. Okay, so this is kind of what is kind of shown here as well, okay? So you have your sister chromatids here after the DNA is replicated, and then you have your other sister chromatids. So two sister chromatids that are red, two that are blue. You see how the breaks are being repaired here by joining the blue to red and red to blue. So this is what we call specifically crossing overs. Okay, so this is how genetic information is actually combined and shared. Okay, now this X formation, as you can see, we call this chiasmata. Okay. Alright, so now let's talk about metaphase a little bit. So metaphase is the next stage in meiosis 1, metaphase 1, and it's exactly what happened like in mitosis. So in metaphase, you have the microtubules, okay, coming out of what? Do you remember those organelles here? So these are centrosomes, okay? So the centrosomes basically produce those microtubules, and they come out basically, and then they attach to the kinetochores, okay, which are located. The kinetochores are, remember, they're protein complex. They're located at the centromere, of basically, these pairs, homologous pairs of chromosomes. Okay. And what do you think is going to happen now? Okay. The next stage is anaphase 1. So the homologous pairs are actually separated, as you can see. Do you see how each chromosome actually have a red and blue combination? So this is due to the crossing over we just saw. Okay, so how is this different than mitosis? During mitosis, if you remember correctly, the during anaphase, right, you have the actual sister chromatids instead of the whole chromosome is separated. Okay, but in anaphase one, uh, the whole chromosome, because we had like homologous pairs here, the whole chromosome is actually separated out like this. Okay, and this is what it's just describing here. Okay, this is why we have a second stage of meiosis, which is meiosis two. In meiosis two, what do you think is going to happen? We want to separate those chromosomes even further, and separate them into sister chromatids. And this is how we have finally the haploid cells. Okay. Okay, so before we get to meiosis 2, let's talk about just the final stage, which is telophase and cytokinesis. Okay, so in the beginning of telophase 1, which is what you're seeing here, right, each half of the cell has a haploid cell or haploid set of chromosomes. Okay, so it's already, each basically half has a haploid set of chromosomes. Okay, so we still have the sister chromatids in those chromosomes, attached with each other because we haven't separated them in anaphase 1. We will in anaphase 2. Okay. Cytokinesis, which is exactly like mitosis, right? In which the cytoplasm is basically, being separated or moving apart. Okay. This basically occurs, and then you have basically the cleavage furrow, which is that little budding here between the cells that separates these two, now what we call haploid daughter cells. Why do we call them haploid daughter cells? Because at this stage, we started with 46 chromosomes here. At this stage, each one of these cells have 23 chromosomes. Okay? Yeah, so it's just showing you here the same exact thing what we just described. So let's talk about like meiosis 2. We'll see, how that finally gives us these 4 haploid cells at the end. Okay. So in prophase 2, prophase 2, right? So we're starting here. Okay. So what's happening is, a spindle apparatus forms, exactly like what we see in meiosis 1 and mitosis, right? So we have the spindle fibers coming out of the centrosomes, and then basically in late prophase 2, okay, um, chromosomes basically are composed of 2 chromatids, right, which the sister chromatids haven't separated here yet. They basically move toward the metaphase plate. Okay, so at metaphase 2, right, again the chromosomes that still are attached, they move to the middle. Okay, exactly like the previous metaphase 1 and metaphase in mitosis. Okay, so they're arranged here in the middle at the metaphase plate. Okay, and so because of crossing over in meiosis 1, okay, you can see how the chromosomes, basically the 2 sister chromatids of each chromosome, are no longer genetically identical, right? I already told you this. So you see how they're different colors, different colored sections. So the kinetochores of sister chromatids, right, which is right here, located at the middle of the chromosomes, attached to those microtubules coming out of the centrosomes, and basically they get pulled apart as what happened in mitosis. They get pulled apart towards the 2 end poles, as you can see in this during anaphase 2. Okay. Okay. So now this is very similar to what you saw where in mitosis. Okay. So in telophase 2, basically, the chromosomes basically arrive at the opposite poles, as you can see here, and now the 2 cells are ready to be separated. Okay, and then basically, the nuclei form, right, to contain the newly formed, haploid chromosome, or basically that's the whole DNA. Okay, and basically the chromosomes begin decondensing. So now, since meiosis is completed, you know, they can just be decondensed and have this spaghetti form again. Okay, so during during meiosis one, right? We have basically we produce two haploid daughter cells. Okay, so here's our first one and here's our second one. Each one of these haploid cell basically goes through this meiosis 2 and gets divided into two haploid um, daughter cells, as you can see here as well. Okay, so total, how many haploid do we get? 4. And this cannot be further divided anymore, as we said before. Okay, and this is just showing the same again. Okay, and again, same thing. Telophase and cytokinesis are the same processes. Okay, you see that we have 4 daughter cells, each with a haploid set of unreplicated chromosomes. So each one of these cells have how many chromosomes? 23. Okay, you guys can see all of these videos on your own. Okay. So this is just the basically showing you the differences between mitosis and meiosis. Okay, so I basically went over all the kind of similarities and differences along the way. Okay, you can just review this on your own. So we know that mitosis basically conserves, whatever we start with, right, the genetic material we end up with. Okay, so it's conserving the chromosome sets. Okay. However, meiosis, right, we have actually a combination of genetic information due to crossing over, and basically we end up with 4 haploid daughter cells here. So you can see that it's actually completely different than this initial diploid parental cell. Okay. Okay, so 3 events are basically unique to meiosis. And all 3 basically occur in meiosis 1. Okay, so why in meiosis 1 is because, this is where crossing over occurs. This is where we call independent assortment. So this is a new word that you haven't heard before, but we will explain what that is. Okay, so synapses and crossing over happens in where? In prophase 1. Do you remember that? Okay, right here. Okay, I'm sorry I'm going through this fast just to remind you where everything is. Okay, let's see here. So basically in prophase 1, homologous chromosomes physically are connected and exchange genetic information, as we saw, right? They're connected to this chiasmata where the X is, and they basically repair those nicks by combining with each other. Okay. And then basically you have homologous pairs are at the metaphase plate, okay, first, and then after that basically the single chromosomes, they get separated into their sister chromatids. Okay, so this is just a summary of both. Just make sure you go over them and study the differences between each one of them. Okay. So let's talk about a little bit, about the variation. What causes variations in genetic information? So mutations is when the basically change in organism's DNA happen, are the original source of genetic diversity. Okay, mutations are what create different versions of genes and what we call alleles. Okay, just remember this. So alleles basically are when genes are being mutated into a it's their own formation. Okay, so this would be the definition of alleles. Okay, reshuffling of alleles during sexual reproduction produces genetic variation. Okay, and the reshuffling happens, in many situations. We will look at them right now. Okay, so um, let's see here. So there are three mechanisms that contribute to this genetic variation, and this is what I was just talking about. So you have your independent assortment of chromosomes. That's the first one. We will see what that is in a bit. And then we have the crossing over, which we studied in just a few slides ago. And then you have the random fertilization. So these three things are what account to producing genetic variation. And that's why, you don't look exactly like your parents. Okay? So let's talk about independent assortment. So independent assortment is also exactly as it sounds. So we're assorting, or we're putting, things at different places. Okay? So this is basically what happened, where it happens in basically it happens like in meiosis 1 initially. Okay, so homologous pairs of chromosome basically orient randomly at metaphase 1 of meiosis. Yeah, this is, I don't know why this is saying metaphase 2, but it's supposed to say metaphase 1 here. Okay, um, so what happened is, after we replicate or duplicate the chromosomes, okay, as you can see here, we have two sister chromatids of each um, chromosome. Okay, we can either separate them, you know, during from going from meiosis 1 to meiosis 2. We can basically put a blue here or a red here, or another possibility, right, which is another type of assortment. Do you see that? We can put a homologous pair here and a red homologous pair here, red and a blue here. Okay, so what does that looks like? It looks like it's a 50% chance of having this and 50% chance of having this. Okay. Okay. So based on these just two possibilities, right, we can create different combinations here, as you can see. So in this possibility 1, we can have basically, since you have during metaphase 2, here we go. So this is, I think, is better. This is a little bit clearer. So in metaphase 2, in which we have two daughter cells after we went through meiosis 1, right, and those chromosomes are aligned in the middle. Okay, we can have basically the blue chromosomes here. I'm just gonna tell you like color to make it easier, and that creates basically, if we're looking at the daughter cells, which are the gametes, right? It's either sperm or eggs. Okay, this gives you basically blue or blue and red and red. Do you see that? Just depending on what the daughter cell has when the sister chromatids are separated during anaphase 2, right? Now looking at the possibility number 2, given a different combination, will also produce a final different combination. After we go through meiosis 2, which produces different daughter cells. Okay. So the number of combinations possible when chromosomes independent assortment is into gametes is 2 to the n. Okay, so where n is the haploid number. Okay, so so for humans, what would that be? So n is 23, we said, right? 2n is equal to 46. Okay, so in this situation, if n is 23, there are more than 8 million, you see that, possible combinations of chromosome. Why? Because when we put 2 to the 23, we're basically getting 8 million. So this is how many different combinations we can come up with. Okay? Now let's talk about crossing over, which we went through it, just a few slides ago. Okay, so crossing over produces what we call recombinant chromosomes. So recombinant chromosomes is kind of exactly like it sounds. It's a recombination, okay, of the homologous chromosomes. So as you can see here, after we went through basically like the DNA replication, we produced a copy of each sister chromatid. We have two sister chromatids. They basically get attached to the homologous pair of chromosome. They get basically close with each other and they create nicks here, and those nicks basically form basically combination uh, of the genes, and that X place is what we call chiasmata. Do you remember that? Okay, and those newly formed chromosomes, homologous chromosomes, they're recombinant. Okay, because they're a combination of different genes. Do you see that? Okay, so basically crossing over contributes to genetic variation, obviously, right? We're combining different genetic information here a lot. Um, so in humans, an average of 1 to 3 crossover events occur per chromosome. So what this is saying is, you see those X's? This homologous pair can actually form two of them, two crossing overs, or up to 3 in each homologous chromosome. Okay? Okay, so, and finally, we went through 3 things here, right? So we talked about independent assortment, we talked about crossing over, and then finally the random fertilization is what contributes to the genetic variation. And exactly as it sounds, right? Random fertilization. So, fertilization is happening at a randomly, right? Uh, you know, we don't know what sperm is gonna basically fertilize what egg, okay? So, that's why like we don't know what genetic information exactly is gonna be passed on, okay? So this is basically creating a genetic variation as well, okay? So the fusion of two gametes, so we're talking about, sperm and an egg, each with 8.4 million possible chromosome combinations from independent assortment, right? We came up with this number from here. Do you remember that? To the 23rd. So this, along with the random fertilization, produces a zygote with any of about 70 trillion diploid combinations. Okay, so 70 trillion combinations. So you can see, each one of us is actually very unique. Okay. So we talked about how crossing over adds even more variation, right? On top of like what you see here, right? So you can see it's a lot of different combinations we can come up with. And I think this is just the final thing right here, talking about a little bit about evolution and whatnot. So this is talking about how natural selection, right, results in the accumulation of, natural selection is selecting for the best genetic variation that's favorable to the environment around that specific organism. Okay. We know by now that mutations are the original source of different alleles, right? Mutations is when we have like basically, differences in the DNA that gets basically repaired, okay? And so these are mixed and matched during meiosis, obviously, right, as we saw. And then you have sexual reproduction contributes to the genetic variation in the population, okay? So this originates from mutations, okay? Um, so animals that are always, reproduced asexually, and they basically produce a copy paste gametes, right, are quite rare. Okay, so sexual reproduction is basically, given a lot of genetic information offsprings. And this is just a summary of meiosis that you're looking at here. So this would be what? Meiosis 1. This would be meiosis 2, which produces 4 haploid gametes, cells. Okay? And this would be the final thing for today. Okay? So I hope you enjoyed this one. So study the differences between meiosis and mitosis. Make sure you know all the different stages. You can see there are similarities, there are differences. Just don't get mixed up between the naming and between what happened each stage. And hopefully this was clear, and I'll see you guys on the next next lecture. Have a good one.